going to have someone be able to explain in like very basic terms, just as, basic as we need it, an explanation of how the neuroscience of memory and trauma works and how the brain works. Today we have Janet Dubinsky, who's been a faithful follower of a member of our team, which we are very thrilled about. Who's going to talk about the neurobiology of learning and memory, a primer with notes on trauma. Professor Dubinsky is a professor in the Department of Neuroscience at our university, and she investigates metabolic compromise in neurodegenerative disease. Specifically, and I'm just going to read this and I won't understand the thing I'm saying, but she'll be able to explain it to us. She researches mitochondrial permeability by looking at how metabolic changes in the brains of Huntington's disease model mice can be used as biomarkers for disease progression and provide us with more insight into the mechanisms of this progression. Additionally, Professor Dubinsky is involved in the creation of the Brain U, which is a professional development program for K through 12 science teachers interested in understanding the brain and its relevance to education. So she's used to explaining things at a basic level for non-specialists. So we are so thrilled to have you. Thank you very much.
Well, thank you. So before I begin, and since we are such a small group, and you now know my name, I would like to know all of your names, and that'll be my test at the end, right, to see how many I remember. So I'm Jan.
Elton.
Elton.
Yeah.
Anna.
My name is Paul.
Paul.
Ophelia.
Ophelia.
Jose.
Jose.
Yasko.
Yasko.
Ophelia.
Ophelia.
Amy.
And Amy. Okay. So we'll see how far I get. All right. But what I want to do is I want to be really, really basic. And if you've got questions as I go through this, I want you to stop me and say, I don't understand that word, explain it. Because the majority of biology is names, nomenclature. So you're all experts in multiple languages, but biology is its own language. And so stop me if I get too confusing. And because I expect you to ask me questions, I'm going to tell you the take-home message at the beginning. And if we don't get through everything, that's fine, all right? So basically what I want to tell you about is that learning happens in our brains because we strengthen the connections between individual neuron cells. Those connections are called synapses, and we'll go over all of this. And as you get chains of neurons connected to each other, they form what we call circuits. All right, so this dotted line is really connecting the synapses to the circuits. And as we learn, we strengthen those synapses. They might have been there from some kind of just, apposition during development, but the actual communication gets stronger the more we practice something and the more we learn something. And as that happens, we actually turn on genes. Remember the genes you get from your mom and your dad? Well, they aren't all on at the same time. They turn on and off throughout your life. And you know that because an infant doesn't look like an adult. All right, a lot of genes had to turn on in order to make that infant grow up. But even as adults, we have genes turning on and off every minute. So there are genes associated with this, what we call synaptic plasticity. And the more we practice learning things, the more those genes turn on and the easier it is to learn. The less we practice, the more we're couch potatoes, the more they turn off, and it's harder to learn. All right, so really simple stuff. And that's really hopeful. But then we have this life experience that comes in.
Can I ask a question? So does this pattern change as you age?
It gets a little bit harder, but it's still there. Okay, so even somebody my age, real wood, where's real wood?
Right.
Has the ability to continue to learn. All right, so it's hopeful. But what I'm also going to tell you is stressful life experiences, some of which might be early childhood adversive childhood experiences, ACEs, or early life stress, maternal absence, or there's one up here. Come join us. Okay. Just as the kinds of trauma that we've been talking about, or the stresses of interpersonal relationships, something temporary. These stresses can have dampening effects in that stress acts at synapses. So it will change synaptic strength and therefore change the operation of circuits, and the stress hormones themselves inhibit gene transcription, which means that some of these positive things get damped down under stress. And so there's some tension here. And that tension sometimes can be handled in a healthy manner, and we call that resilience, and sometimes it can't be. And then we have people who really collapse or develop pathologies under the impact of lots of stress and stressful events. So that's the take-home message. All right, I'm going to go back now and I'm going to fill in a lot of details. So stop me if the words get complicated.
I have a little thing about the genes part. I know the genes have the program that if some part of tissue gets damaged, they have the program that replaces them. Now, does it also influence the way you think about things? Well, I'm thinking mechanically. It's a program. So it's active all the time, or in some cases not. Like a certain gene can determine the color of your eyes, and another one it can make it change. So you turn it on and off, and then another one replaces it. And
so things like the color of your eyes don't change after a few months of life.
After they're established.
After they're established. So, my hair will be dark until the gene that makes my hair dark decides to stop, and then it's going to go gray. And it's started, but it's not all the way there yet. Okay, and that happens in blondes sooner than it does in brunettes. People go gray here in their 40s and 50s because they're Scandinavian blonde descent, as opposed to those of us with more depth to our color. All right, but there's a gene that makes the color. That produces that color, and it sometimes in life decides to say, I'm tired, I'm done.
I know there are some chemicals, some products, maybe started by the genes, that when you don't have them, you don't have the melanin.
Right.
It's like
but the genes I'm talking about are not going to be something you're going to see in an external feature. The genes I'm talking about are going to be for proteins, which are the building blocks of everything, and they're the workhorses. They're the engineers, if you would, or the engines of what goes on inside a cell, and we're going to talk about a lot of them. They have names, okay? And so I'm going to talk about genes turning on and off certain proteins. All right, so I hope to build you a better picture of this machine, okay? All right, so the other thing I want you to see is I really, really want you to see a real neuron. So this is a real neuron in the cerebellum, which is the very back of the brain. So I brought a brain. Can everybody see it? All right. So the cerebellum is this part in the back. So this, sits in your head, everything from the ears on up, and the spinal cord. So the neurons in the cerebellum are very stereotyped. And this one, they're actually gorgeous. This is called the Purkinje neuron, after the neuroanatomist Purkinje. And what you see is the cell body, and the tall, green, windy piece is the dendrites. That's the input to the neuron, or the dendrites. It has an axon, which you can't see, but might be here going that way, which is the output. So when we think of neurons, we think of them as having directional input and output with these processes. All right, and there's usually only one axon and lots of dendrites, but the axon branches. So, it's a branched input and essentially a branched output. And the green is an axon coming from far away in the brainstem. So the cell body for that red axon is somewhere way down here where my fingertips are. Okay, far away from here, several centimeters at least. And that axon is going to climb up that dendrite, so it's called a climbing fiber, and every place you see it get a little bit bigger in imagination, a little red dot, that's where a synapse is. So the axon is presynaptic to the dendrite, which is postsynaptic. And at each of those synapses, communication occurs. So now you've seen a real neuron. And if you went to the Cajal exhibit 2 years ago at the Weissman, you saw plenty of them. Okay. So how does communication occur? So here we have two neurons, the first one on the left and the second one on the right, and they're going to talk to each other. There's an electrical signal that the neuron forms to send its message, and that electrical signal travels down the axon. And when it gets to the end, what we call the nerve terminal, it can't go any farther. The electricity is confined to the inside of the cell, and it can't go across the space. So that electrical signal is changed into a chemical signal, and the chemical signal crosses the synapse and is tasted by a protein called a receptor on the dendrite. So we've got electrical in the cell, chemical between the cells, and then that protein receptor forms a new electrical signal which comes into the cell body of the second cell. Now those cell bodies are big adding machines. They add up all of the incoming information and they decide if it's important enough to send the message further. Because they're not only going to get input from one particular place, but they're going to get input from another place. So lots of these synapses on dendrites send information in, and if they all arrive importantly enough at the same time, that cell body says, ooh, something's going on, I better tell the next neuron. And then it sends an electrical signal down its axon, and this is what we call a circuit, when you get this movement of electrical to chemical to electrical to chemical to electrical to chemical. Now those chemicals we left sitting out here in the extracellular space, they can't sit there forever, because then the postsynaptic neuron will just be saying, oh, I get lots and lots of input forever. And that can be overwhelming, it'd be too much. So that those chemicals have to be recycled. So they actually get taken up by the nerve terminal. So they're basically pumped back in. It's a slower process, but it clears the extracellular space of the trash. So the trash has to get collected and recycled and reused. And now it's ready to do the same thing all over again. And when this information from neuron 1 to neuron 2 keeps going on and on and on like that, we call that excitation, because neuron 1 is telling neuron 2, do something. I'm exciting you, you send this message on. And that depends on the flavor of the chemical that goes between the two. All right, the flavor of the chemical, so that's excitation. If we change the flavor, we can change the message. So this time, I'm going to change the flavor, and this actually has to be a separate neuron. One neuron doesn't change its flavor. But this illustration, notice that the chemical is a different color. Since it's red, and guess what message it's telling the second cell? Pardon?
Danger?
Not quite danger, but it's basically saying, stop. It's saying, don't send messages, all right? So if you think of the excitation as a +1, okay, think of this danger stop signal as a -1. And then the cell body has to add them up, and it says, oops, nothing's going on that's terribly important right now. So having got that inhibitory signal, now information coming in from elsewhere adds up to it, and the cell says, nothing's going on. No signal gets sent. And so we call that inhibition. And if you notice, even the inhibitory neurotransmitter got recycled. All right, so this is the basic process of synaptic transmission, where the electrical signal that goes on within one neuron has to get translated to a chemical signal sent across a small teensy space, and then becomes a new electrical signal and gets processed by the second neuron. And this happens over and over and over again throughout your brain. Question?
Since you invited us to ask questions.
Absolutely.
So, like, for example, like babies or young kids, do they have a whole bunch of these things happening, and then you said that like there has to be some kind of mechanism to determine like which are important and which aren't, like.
Yep.
So as they become like socialized in one way or another, then do certain of these messages become like told that are not important and they're not picked up or something?
Like the infants have to figure out what the world looks like.
Right.
They don't have any preconceived notion of visual space because they've been living in a womb with their eyes closed. So all of a sudden, they have to make sense of these blobby images of their parents' faces and maybe siblings, constantly in front of them. And so they begin to distinguish dark and light, and then they begin to distinguish shapes and faces, and then they have other sensory input that comes in, but they begin to extract the signals of the world around them from the background noise. So there's a constant learning of what the world looks like, and then finally they smile because they recognize the world, or they recognize players in their world, and then they begin to interact with their world. And so yes, there's a tremendous amount of synaptic plasticity going on in that first year of life and for the first 4 or 5 years of life, and even through K-12. Okay, when you get to be my age, you want there to be plasticity, and you hope there's plasticity, and we work to maintain that plasticity. But it's not nearly the same as in the first few years of life, absolutely, because that brand new nervous system has to figure everything out. But you don't have to figure out what the world looks like now. You know what the world looks like, and you can ignore the walls around us. We're in the room concentrating on talking to each other and looking at my slides. So you're way past some of that very early learning, and you're thinking about much more complex ideas. So your learning is taking place at a very different level than an infant's learning.
This electrical signal going from the axon right there to the dendrite, I thought that releases one neurotransmitter chemical.
The chemical is called
But it's always the
neurotransmitter.
same, right?
This one neuron will release one neurotransmitter. A different neuron will release a
Would be
different
released by a
flavor.
different axon.
Yeah, and I was lazy. I didn't redraw my
I
neurons.
thought that
No,
it
no,
has the ability, we have to be coded that saying, when you get that signal that way, you create this, when you do it that way, you do that. That's not the way.
No, one neuron, one flavor.
Sure.
And I was lazy. I didn't change my picture. Okay. All right, so let's think about what's going on. That's a nerve terminal. There's an electrical chemical signal. It causes an electrical signal in the dendritic spine, which is C. Everybody follow that? Mm-hmm. All right, so what happens if we send in signals on 2 nerve terminals, coming from nerve terminal A and B? Come on, there we go. We get more chemical neurotransmitter, taste more receptors, we get a bigger signal. We do that often enough, that bigger signal is a stronger synapse. So the cell learns that more important information is coming in because it's got more input. All right, so if it gets enough practice with that information coming in, what can happen is that synapse can split in 2. See, we now have 2 synapses here. And each of these little bumps is called a spine, a dendritic spine. So those are structures that we know are where the synapses are. But yeah, if we go back, remember there was only one synapse initially, and it got so good and so strong that it split in 2 and made 2 synapses. And in order to split in 2, it needs more proteins, it needs more of these receptor proteins, it needs more structure, it needs more good chemicals inside, and in order to do that, it had to turn on genes to make those new proteins to make the second synapse. All right, so that's the kinds of proteins we're talking about turning on. Small ones that you can only see at a microscopic level.
Is this what they mean when actions that fire together enhance memory, or
Yeah.
is this what?
Firing together, you wire together.
Is this what this?
This is exactly
Signal
it.
is stronger.
Absolutely. All right. So, yeah,
And
question.
I have a question. So before you mentioned the excitation and the inhibition, so do you have, in that example that you provide, is both of them are either excitation or inhibition? It can happen that one is sending one message.
Oh,
and the
yeah.
other one
I did-
is
so-
sending the contrary message.
It could be. These could be opposite, sure, but I'm only working on excitation because that's additive and strengthening. If one's additive, with an inhibitory one, then there's not going to be any strengthening because they're competing.
Okay.
But you could have two inhibitory inputs, and they could strengthen each other and get stronger inhibition. So, yeah, there's valence and then there's process. But you can strengthen in either direction because we get lots of complexity. But what about this synapse here? We haven't seen any activity on it. Any guesses what's going to happen to it after a while? Yeah, it dies. The cell says, why should I be pumping protein into this little dendritic spine here? It's doing nothing. Let's put that protein someplace else and that spine goes away. It retracts. So at this microscopic level, there are changes going on that require genes to turn on and off.
That's why we have actions and evolution.
Sure. All right, so what we think about when we talk about the whole brain and not just synapses is that we think about the hippocampus, which is this green structure right in here that I'm now covering up, which is not actually on the outside of the brain, it's actually deep in the brain. So if I hold my brain like this, the tips of my fingertips, deep down inside this model brain is where that hippocampus is. So that picture is deceptive. It's supposed to be showing you what's going on inside. And I don't like these ones where it pretends it's on the surface. So the hippocampus basically is gating information that comes in and out. And it's the place where we think about short-term memory residing. And if something's important enough, then it goes into cortex, which is all of this top coating, the top surface of the brain that's so convoluted. And if we take slices of hippocampuses from rodents and other animals, we rarely get them from humans. This is what a slice looks like, and you see a very stereotyped circuitry. We've got some inputs, which are the red axons coming here, and we have different, that's one input. And then we have, each one of these blue triangles is a cell body. And in this particular part of the hippocampus, these are the dendrites, so here's inputs, and then there's outputs. That's an axonal output, and this is an axonal output. All right. Oops, that's not what I wanted to do. I wanted to erase so you could see the picture. No, sorry, I don't want to erase it. So this axonal output from the CA3 pyramidal neuron, synapse is on the dendrites up here on the CA1 pyramidal neuron, and we can stimulate with these black wires those axons, and we can stick electrodes in that field of dendrites, and we can record the strength of the synapses electrically. And what we get is, for a very low-level stimulation, once every 20 or 30 seconds. I mean, something really very boring. All right, we get a basal level of synaptic strength. But if at the time of the arrow, we give a barrage of stimulus, all of a sudden we go, I mean, we really zap it. We're not simplest scientists. We really try to maximize the system. All of a sudden, synaptic strength increases a lot. And depending on the characteristics of that zap, whether it's really strong enough or not, that synaptic strength may stay elevated for a relatively short amount of time, here 60 to 80 minutes, or it might stay elevated for as long as we care to record. And we've gone out for days. All right, so this change in synaptic strength from baseline to this elevated level we call long-term potentiation. And that's what I mean when I say synaptic strengthening. Okay, is that we've changed something on a relatively permanent basis, or as long as we care to keep that particular piece of tissue alive.
I assume you cannot get a precise, this is, in general, I suppose it's like going with a hammer. I mean, you cannot pinpoint, right?
Well, this is the sum of many synapses simultaneously. People have looked at individual synapses, but not this particular recording. This is a relatively old experiment. We've been doing LTP now for 40 or 50 years. So this is well established. But I think you need to see it because that makes what I'm saying not just a cartoon, but real concrete. Now, this synaptic potentiation, which we call plasticity, is synapse specific. If we stimulate the input on the left, those blue synapses will strengthen, but not the synapses from the red neuron axon terminals coming in on the right. Unless they're both active at once, and then the simultaneous activation acts instead of the barrage. Instead of me zapping it, simultaneous activation or associative activation says, go neuron, and then the synapses will strengthen because they are mutually reinforcing each other. Question?
When you say you stimulate it, how do you stimulate it? When you say you zap, are you talking about electrical?
Yeah, I'm talking about sending a current through this particular electrode that's in the prep. So when we do these experiments as scientists, we are not talking about a sensory stimulation. Now, we are more sophisticated these days, and we can put electrodes in brains of animals and do real sensory stimulation and record, but not at the level I'm showing you here. Okay, I'm just showing you some real basic stuff. And the persistence, the question is what's going on in that persistence for a long time? Well, there's lots of mechanisms, and basically we can change the proteins. We can increase the number of receptors. We can increase the amount of chemical that's released. We can make long-lasting changes in the intracellular chemical environment, which requires protein synthesis. And you don't get new protein synthesis without turning on genes. And that, so you see where I'm building my story here? All right, so this is a breakdown diagrammatically of the presynaptic nerve terminal here at the top. So you have to pretend that there was a nerve, an axon coming in, and this is the little button or bouton at the end. And here's the dendritic spine. And you've got to remember that it bumped out of a dendrite that was somewhere along the bottom here. All right. And now I'm blowing things up so you can see the individual molecules, the individual proteins that are tasting the neurotransmitter. The neurotransmitter here is glutamate, so GLU, these little funny shaped black things. And the glutamate has 3 different flavors of receptors. One, it just fits into the receptor, and that protein then starts a chain of events with other intracellular proteins that eventually raises intracellular calcium. The other 2 receptors, let's look at this one over here. This one, when glutamate binds to the receptor, when this bump fits in the hole, so it's a lock and key like mechanism, then that receptor actually has a hole in it, and it undergoes a conformational change, and the hole gets bigger. So when the hole is small, water can pass through, but that's about it. But when it undergoes a conformational change and gets fatter, then you can get small ions to move through. You can get sodium and potassium to move through. All right? And sodium and potassium each have a positive charge. And so the movement of that positive charge causes a current. And here you can see the sodium and the potassium moving. And the movement of that current is what's forming the new electrical activity. So we went from electrical to chemical, because basically the chemicals are inside these little vesicles and they get spit out. I didn't tell you how that happens, but don't worry, it happens. They get spit out, and then the glutamate crosses this space in between, binds to the receptor channel opens, and as a consequence of depolarizing this first receptor, the second receptor performs a minor miracle. The second receptor, the hole was a little bit bigger to begin with, and there's not only sodium and potassium running around, but there's calcium and magnesium running around. And calcium's on the outside, a relative amount. And on the inside, just a teensy amount. So when a hole opens up that's big enough for calcium, calcium's going to rush in. All right, and that's true of the sodium and potassium. But in the unstimulated state, this particular channel likes magnesium to sit in that hole. And the magnesium's too big to get in under any condition, so it blocks the hole. So the first receptor has to allow current to flow, so the inside of the cell becomes a little bit more positive. And that making the inside of the cell a little bit more positive, the positively charged magnesium says, oh, this ain't where I want to be. And the magnesium hightails it out of there. And then when the magnesium's gone, then the calcium can go through. and raise intracellular calcium. So there's a delay for the second receptor, and that delay is what you need in order to learn. If just the first receptor opens and calcium never changes inside the cell, the synapse is the same strength it's always been. But if you wait and there's enough stimulation here, so you get enough of the first receptor to open that you pop out the magnesium and the calcium comes in, then because calcium was so small inside the cell, such a low concentration, now the calcium can act on other proteins inside the cell and turn them on. There's a whole slew of calcium-activated proteins that bind calcium and they do something. Wouldn't you like to just taste a little calcium and do something? Okay.
Is this the same calcium that we take in our pills, like in our
Absolutely.
supplement?
Same calcium that goes into your bones, stored in your bones, absolutely. All right, calcium's calcium. Anyway, so there are all of these calcium-activated proteins we call protein kinases. And those protein kinases actually add a phosphate group to the first kind of receptor, and enough of those happen, more receptors go into the membrane, and that's how we get strengthening permanently, because we've changed the amount of receptor. And those protein kinases hightail it down to the nucleus. So they go back through that axon all the way back to the cell body to the nucleus to turn on genes. And that's where you get the genes turning on to form the new proteins you need to build things. All right, now, before I go on, any questions? That was a lot, yeah.
No, so what's the time?
This?
Yeah, how fast does this happen?
Well, the synapses happen pretty fast, on a millisecond basis. But
And
the
then
learning, the calcium bit, that's a little slower. That's going to happen on the order of hundreds of milliseconds, maybe a second. A little slower, okay? That's still pretty fast, yeah. But to get the protein kinases and the genes turning on, that might take you hours. So every step of the way here has a different time scale.
So, the transmitter send whatever is coming out there, but it's controlled by the receptor. The receptors determine the diameter. They allow, so you have two things. One, a transmitter sends a signal, and the other one changes because some signals are necessary.
When the neurotransmitter binds to the receptor, then it changes the conformation.
So the transmitter only says, I send this, if you need it, you regulate it.
That's right. And it's somewhat random because the transmitter's just kind of floating around in this space. It's an aqueous space, a water-filled space. So there has to be a lot of it to make sure some of it gets to the receptors because some of it's going to go off in some other direction. It's just
So
a
it
random.
comes from the spine.
Yeah, it will, and after it's bound, then it has to unbind, and not only does it have to unbind, it has to get taken up again and repackaged, okay, or wander off too far to get repackaged.
So if for some reason you had like not enough calcium, like let's say like menopause, if that does that then affect, is that one reason why maybe at a certain time you may be creating less of those things and maybe you don't learn as quickly
Calcium
or
is
something?
really tightly regulated. Calcium, no, these are all important. Calcium is really tightly regulated. So, we should have about 2 millimolar calcium on the outside and 10 to the minus 4th millimolar, or 10 to the minus 4th millimolar calcium on the inside. So, 10,000 times more out than in. And there's hormones in your body that maintain that calcium concentration. And they do a lot, because the most of the calcium's in your bone. So they're mostly making sure it's stable in the blood and extracellular space and extra stored in your bone. So I'm not going to worry about the amount of calcium in your brain at any one time. You would your electrolyte balance would have to be really wacky for the calcium to go off. All right. There's some concern about calcium in the heart, but it's really more the sodium and potassium that are the bigger concerns in terms of electrolytes. Calcium is very tightly regulated, but yeah, we need it for bone density as we get older, because other hormones are interacting with, the lack of estrogen is the bigger issue here. because that's causing the calcium not to be stored as much in the bone. So it's a different system. All right, so what goes up has got to go down. Not only can you potentiate synapses, you can depress them. Okay? So instead of giving a huge barrage of activity, experimentally, we give them a really boring stimulus. We give them a stimulus once every second. All right, and the cell seems to get a different pattern of calcium and does different things. But I want to show you a real experiment really quickly. This is a mouse that has been trained to sit in this tube and to get water from this little water port here. So it licks the water port, and it doesn't get water for a couple hours before the experiment, so it's thirsty when it gets in there. And so it knows to drink. And that we've taken off the skin and thinned the scalp. We've just scraped away some of the skull, so we can put a microscope up against the top of the animal's head, and we can see the neurons inside on the top of the mouse's cortex. And this mouse is a genetically altered mouse that has a calcium-sensing dye in the neuron cell bodies. So we can see when the calcium goes up. So instead of recording the electrical activity, we just look for the calcium activity with a microscope. And the animal's head is actually fixed, so the microscope doesn't have to move with it. So the microscope's fixed and the animal's head's fixed to the microscope. And the mouse is terribly happy to sit in there and lick for its water. All right? They crawl into these things very, very, very happily. So this experiment, the mouse has already learned that there's a light cue that goes on. So this red thing is the light cue. And then it gets water, and then it licks for water. So it knows to do that. But the new part of the experiment is there's a lever this time. And the mouse has to learn to press the lever to get the water. Okay? So when the mouse learns, you can see the lever that it can learn to press the lever. That's this trace here. Okay? So now we're going to watch what happens as the mouse learns. And you can see on the first day, the first trial, maybe there was one press of the lever accidentally. Didn't get a whole lot out of it. But by the 5th day, there's a little bit of lever pressing going on. And by the 2 weeks, the mouse has got it down. Every time he gets in, he knows press the lever for water. All right. So, it took him a couple of weeks, but he got it. All right. So now let's look at what the microscope saw. On the first several days, every different color represents a different neuron that is visible on the surface of the mouse's cortex, and it just happens to be motor cortex for the mouse's paw. So it's very convenient that we can see the mouse, the neurons that are controlling the paws. And so on the first few days, there's a lot of scatter, but not a whole lot of real synaptic activity. 5 to 7 days of training, and all of a sudden you're beginning to see a lot of activity timed to when that light cue goes on, and the mouse knows to start pressing the lever. And by the end of the training period, look at how synchronous the firing is of all of those neurons. You could see the recruitment of the neurons into the circuit that says, press the lever for water. So we are changing synapses and changing circuits as we learn, and this is happening across the animal kingdom.
Can you talk a little bit about the scattering? There's a kind of a line in the middle of the colored dots. So they're all bunched together there, and in the 12 to 14 week one, it's even clearer, so that stops there. But what's going on below that?
So what you've got is for all the neurons that the microscope saw, each one, different colored one, they ordered them top to bottom between the ones higher up at the top are the ones that responded closer to the onset of the stimulus light. Okay, so when the cue light goes on, they just, the top neuron, which is a green one, here, and there's a purple one on this side, turned on immediately to the light. Whereas down here, these neurons have a greater lag between the time the light cue went on and when they start to fire.
So what causes that?
Their place in the circuit. You're not giving, it says, look at, that's a time scale is a second. So if I told you that a millisecond or two is the time it takes for a synapse to fire, there's a second delay between here and here. That means that these neurons are very downstream in the circuit. There are 1,000 synapses later. We're not talking about which neurons talk to each other here. We're just saying where they operate with respect to the cue light. And so there's a lag time. So they could be part of a feedback circuit. Rather than the one that says press. We have no clue what each individual neuron is saying. All we know is that they are part of a circuit that has been learned to press the lever. That's all we know in this. But it's real learning taking place. All right, so we have these protein kinases turn on genes. How is it we have individual memories? Think of this grid as dendrites in the vertical lines and axons in the horizontal lines, and every possible crisscross here is a potential synapse. All right, when I was a child, we went to my grandmother's house for dinner every Sunday night, and we always had roast chicken, without fail. So I have one set of synapses that remembers my grandmother's house. And I have another set of synapses which remembers chicken dinner. And some of those overlap. My grandmother hasn't been with us for decades now. But I still myself make chicken dinner. So sometimes when I make chicken dinner, the chicken dinner is purple, so here's when I make chicken dinner, you know, it just as a chicken dinner memory. But sometimes when I make chicken dinner, I think how nice it was that I had chicken dinner from my grandmother's house. And activating the chicken dinner synapse brings up the memory of grandmother's house because they were shared synapses in making this memory. So not every synapse encodes every memory. It's the sparse coding of a few synapses in some places scattered throughout this entire big brain of ours that encode one memory and a different memory. And sometimes we talk about our wires crossing because indeed the synapses might be shared between multiple experiences. And you don't need to activate the entire set of synapses to recall the entire memory, just a subset.
So is that where synesthesia comes from, where you can smell color or?
Synesthesia comes from the fact that these axons branch and they may be going to parts of the brain which have different sensory encoding going on in them. And it could be wire crossing, yes. But not everybody has synesthesia.
No,
In fact, most people don't, so.
but we all have it in literature.
Of course, of course, we can imagine lots of things. And that imagination, that creativity is the crisscrossing of synapses that didn't make sense until you wrote a story about it. Okay, so yes, I think that a lot of this randomness, because there's a lot of randomness in how all of these synapses work, is part of that. And I started to write, I didn't really want to, but we have 86, 1, 2, 3, 4, 5, 6, 7, 8, 9, 86 billion neurons in our brain, okay? And each of those has at least 1,000, if not 10,000 synapses. So if you want to talk about, this is be neurons, but then you add 3 more zeros, you get 86 trillion synapses. In your brain, you have as many synapses as there are stars in the Milky Way. Who knew? So that's a lot of crossings. A lot of wire crossings. All right, so much for the memory. Come on, let's go. All right, so we're back to where I started in terms of learning and memory and synapses and circuits and turning on genes. Everybody with me? Questions?
Yeah, I mean, you're talking about synapses as things, and I've always thought of them as events.
But they can be both. Okay, so I'm looking for my picture there. Okay, so actually, we think of the synapse as being the presynaptic nerve terminal, this space we call the cleft, and the postsynaptic spine. So all of this together is the synapse.
So that's a thing.
Well, but this is space.
Right.
You know, it's this apposition and it's all of this molecular machinery for spitting out the neurotransmitter and tasting the neurotransmitter. It's the conglomerate. So it is also the event of communication across that space. It's a hard concept. because it's not just the cleft, and it's not the pre, and it's not the post, and it's not one thing, it's everything working together. It's the whole mechanism.
So you can get signals that say, do something, and only one that don't do it. So it's like a calculator kind of thing.
Absolutely.
You add, subtract.
In fact, when we've got a neurotransmitter that says stop or inhibit, it's just a different flavor. You know, I might give it a name GABA, because that's one you might have heard. But the receptors act the same way. But instead of passing sodium and potassium, they pass chloride, a negative ion, and that adds, that subtracts instead of adds. So it depends on, again, valence. And there's just tremendous detail in all of this. And it goes it gets really complicated, but let's go beyond, because you want to talk about stress and how stress changes all of this. All right, so we had to get the basics down. All right, so it's good that we have a little bit of stress in our lives. All right, stress is motivating. You know, you want to finish that chapter. You want to make that book, or that story, or that short story, whatever it is. And you know, your memory needs a little bit of stimulation too. You know, I need to write the story for my daughter's birthday, whatever. But too much stress, and it can be debilitating. So we think of a response to stress, and here I'm actually talking about the stress hormone cortisol, which is of a class called glucocorticoid. So you're going to see glucocorticoid a bunch. So that's just the class of chemical. And as you increase the amount of stress hormone, you can go from this relatively healthy range into a less healthy range. All right, and it's the same curve basically. And I'm not sure if you can read any of that. I'll just point out some of it. But the synaptic function for low amounts of cortisol and reasonable amounts of stress or challenge can be very adaptive in that you can, you know, you get long-term potentiation and you build synapses and you build learning. And that also, you know, happens in short time scales. So here the axis isn't exactly the concentration, it's actually time and how long and persistent the stressing situation might last. But, you know, for a short one, a short amount of stress might be adaptive. You might, say, I did get that wrong, but I'm going to learn from it, and I'm going to go back and fix it and do it again. Okay, you know, maybe you're painting and you'll paint over whatever didn't look right. But you had some visual feedback, you went through a process. If the stress becomes chronic, longer amounts of time, then that adaptation might not work so well. There might be give and take, you might be able to manage it for a while, it's short term, but eventually you can get, in the months to years amount of stress, it can become damaging. And the synaptic transmission will actually suffer. And so will long-term potentiation. You'll have less of that. And it'll be even worse, we sometimes birth new neurons. Not a lot. I mean, compared to this number, new neuron births are basically 100 to 1,000 per day. And there's more when you're younger than when you're older. So, and they only happen in that wonderful place, the hippocampus. And it's mostly to replace the neurons in your nose from smell, because they die off from the chemicals that come into your nose. But sometimes they happen to go into the memory pathway too. And so we like these new neurons, but there's not a whole lot of them. But as we get older, you can get less of them. And stress can decrease that rate of new neuron birth as well. And we consider that a loss of resilience. So that there may be a lack of ability to compensate, that we've gone beyond the ability of the system to manage itself, and we get these damaging effects. So we can think about stress not only in terms of increased stress hormone, but also in lengths of time, and the changes that accrue with time in this process of getting the synapses to work. so now I have to back off and give you a little bit more about the stress system itself. I talked about the stress hormone cortisol. Cortisol is made in the adrenal gland, just sits on the back of your kidneys. All right, so we're nowhere near the brain, but that's where this cortisol hormone comes from. And it doesn't get secreted regularly. There's a diurnal rhythm that I'll show you, but it also only gets secreted when there's a signal that tells the adrenals to do that. And that signal is another hormone called ACTH that comes through the blood and gets to the adrenals and tells the adrenal it's time to secrete some cortisol. Where does the ACTH come from? The ACTH comes from a hormone in the pituitary, sorry. And it's not shown in this picture, but the pituitary is a teensy little pea that sits right about here. I mean, I don't even have one on this model, but way at the very base of the brain, just above the back of the roof of your mouth. The little pea, and that pea is the pituitary gland. It's actually not nervous system at all, it's hormone system. And it secretes the ACTH, but it only secretes the ACTH when the hypothalamus, which is a part of the brain that sits just above the P of the pituitary, secretes some hormones, cortisol releasing hormone and arginine vasopressin. So this diagram's not quite anatomically perfect, but it's showing you kind of where this is all happening. All right. And what causes the hypothalamus to secrete the releasing hormones is signals from the rest of the brain saying something's going on. All right? It might be an arousing emotional stimulus. It might be a thought process. Oh, I'm remembering that my dad just died, and so I'm stressed. So lots of things. Or it could be a stressful external event. I was in a car accident. Or, heaven forbid, one of these more traumatic kinds of incidences. That cortisol has two main actions once it's in the bloodstream. The first one is very short term, and it feeds back onto the brain, the pituitary, at various places, and that cortisol binds to its receptors in those various structures and turns off this cascade of hormones that's telling the adrenals to make cortisol. So it inhibits itself. Okay? It says, all right, I've gotten a short enough signal out here. We don't need to continue to send out stress signals. So there's a short-term inhibition. And this one's taking place on the order of minutes to hours. Okay? So it takes 2 minutes from the time the brain decides to release one of these releasing hormones until the adrenal gland has spit out some cortisol. So it's a slow process because all of those hormones have to go through the bloodstream to get to the adrenal and get there in enough concentration to do their thing in the adrenal. And then the adrenal gland has to say, oh, I'm going to manufacture the cortisol because it's not sitting around in the store ready to spit out. It actually has to be biochemically manufactured before it can be spit out. So there's delays in this process. That's the short-term response, is to just regulate itself. The other thing the cortisol does when it hits any cell, anywhere in the body, and this is a generic cell here, and this is a generic nucleus, this double membrane, the nucleus. The cortisol binds to a glucocorticoid receptor, a special receptor just for the cortisol, and it binds to other proteins and crosses into the nucleus, and where it binds to DNA. And it is a signal that turns on transcription of that DNA and makes it into RNA and begins this process for getting new proteins. So it's genes expression. It's turning the gene on, basically. So the stress hormone can turn on genes. And that takes, biochemically, several hours. All right, it's a slow process. Biochemistry is much slower than electricity. And here it's turning on a gene for a protein with this horrible name, FKBP5. I can't even say it. But this FKBP5 protein that is now made several hours later, turns out it goes back and it binds to the glucocorticoid receptor, and it tells the glucocorticoid receptor, don't go into the nucleus anymore. It's time to stop telling genes in the nucleus to turn on. So there's a feedback signal here too. So not only does the glucocorticoid receptor activation turn on a lot of genes, it also turns on this one that turns around and turns itself off. Okay, biology's full of loops like that. And that's good, because you get a short signal, you don't get a continuous signal. All right, so we have short-term and we have long-term effects of cortisol. And I'm going to tell you more about the cortisol, or we can stop. But I do want to say that the cortisol is a short-term stress signal. What Brian was talking about in your first lecture was other molecules that signal chronic levels of stress. So he was talking about a totally different system. And I'm not going to go there at all, since he talked about it and knows much more about it than I do. But I want you to get the sense of the multiplicity of these regulatory mechanisms.
And what are the genes that get turned on by the cortisol? What are those genes? Are they specific genes? Do they have specific functions?
Yes, they do. And what do you think let's make some predictions. What are my favorite functions?
Well, I mean, if so, they're turned on by stress, so are they negative for the body?
Well, sometimes yes, sometimes no, but yes, they will turn on lots of different genes. The glucocorticoid receptor activates lots of downstream genes. And I'll name a few as we go on, but some of them are these receptors. Some of them are these synaptic plasticity receptors. So it feeds back on the nervous system and changes our ability to have plastic synapses by regulating the proteins involved in synaptic transmission. Yeah, so everything's loopy.
Yeah.
The nervous system is full of all of these feedback loops. And I don't know, I've been talking for an hour. You want me to keep going or you want to stop? Your choice.
Give it back.
Yeah, keep going.
All right, so basically, this is just a graph to show you that the beginning of your active period for us, which is daylight, we get this and circadian rhythm and glucocorticoids secretion, cortisol secretion, but we also have hour-to-hour, moment-to-moment fluctuations. So we have these two competing rhythms, but there's this overarching daily rhythm. And then we remember stresses. We remember stresses because of the stressful event, the car crash. It was immediate, impactful, and we remember all, the truck that hit us or whatever it was, or we were rear-ended, because the synapses had a very strong emotional activation at that time. But the stress signal came 3 to 5 minutes later, once the cortisol got going in the system. And then, once the tow truck's gone and everything's all cleaned up, and then we say, oh, and you feel shaken like this for quite some time afterwards, because our body is still dealing with these many hours long processes. And the question is, what happens to those signals? How do we get signals that cause memories to change? And it turns out we spend a lot of time looking at rodent behavior, and one can stress a pregnant rodent dam and then see the same effects of that stressful signal on the pups later. And the question is, what's happened? How did the pups learn if they were in utero when the dam was stressed? And one way would be that this cortisol that went up in the dam system when she was stressed crosses the placenta, and it does that. It's capable of crossing the placenta. So that's a reasonable way, and then would activate the pups, what we call HPA axis, hypothalamus pituitary adrenal axis. So this whole loop is the HPA axis, because it involves all three of those structures. And a pup will be learning basically how to express those glucocorticoid receptors, and stressing the pup would change the amount of glucocorticoid receptors that were then expressed and put into the membrane of the cells in the pup. So that could be through the HPA axis, but the pup could also learn through some kind of a genetic or heritable form of transmission. And so people began investigating that, and they begin to look at the glucocorticoid receptor expression. And here's the experiment. Turns out that dams, rat dams, have two different kinds of behaviors in terms of mothering behaviors, parenting behaviors. Think of the good rat as licking her pups a lot. And so we have a pup, or a rat mom who's licking her pups constantly. And then we have more nervous dams that don't lick. They just let the pups go. You know, they eat and they nurse, but they're not really attentive to the pups. You can see any rat in a litter, or with their litter, and tell the behavior pretty quickly just by watching. So it turns out that if you then look at the pups' brains when they grow up, the pups from the litter that got licked, they have lots of glucocorticoid receptor in their hippocampus. Whereas the pups, the grown-up pups from the mom who wasn't so attentive, have many fewer glucocorticoid receptors. Now, why did the gene expression change? Is this an instinctual behavior that some rats have and some rats don't? Is this genetically determined, just like eye color is determined? Well, we don't know the answer at this point of the talk, but I'm going to do an aside. I have to take you back and talk about gene expression a little bit more before I can. I'm going to dangle the story here, all right? We're going to go do an aside.
There's a question.
Yes,
The
question.
concept methylated. I know it's very important and related to many things like cancer and so on, but I have no idea what it means.
I'm going to get to what that methylation means. I'm going to explain that. That's my aside. Okay, so yes. And the other piece, not only the rat pups, or the grown pups who have fewer glucocorticoid receptors, the genes had lots of methyl groups on them. So you notice that in the slide. And those grown pups who have lots of glucocorticoid receptors have very little methylation on their genes. So the question is, what are the methyl groups doing on these genes? Okay, that's the biochemical question here. But I have to explain a little bit more biology. Remember, this is basic high school biology. We've got genes, and the genes, which are DNA, double-stranded, you have to open up the two strands in order to transcribe those into RNA. Okay, they open up, they get transcribed into RNA, and then the RNA leaves the nucleus and goes into the cytoplasm, where it's called messenger RNA, and then it's translated into protein by adding amino acids together, okay? That's the basic way we get protein out of DNA. But the DNA, and here, everybody can have a piece of string if you want. You can have a piece of DNA. All right. The DNA is twisted, it's a double helix, and if you twist that double helix enough, and you keep twisting it enough, basically, it forms balls. So everybody can twist their DNA, and you can get it to just keep twisting. It'll just curl over on itself and form these tight little balls. And that's what we get when we talk about our chromosomes, which is all of this DNA that's so tightly wound, so tightly spun onto itself that it forms these teensy, teensy, teensy little tightly wound balls. And the question is, how do we get the DNA out of those balls when we need it to turn on a gene, especially one gene in the middle of something? All right, so biochemically, that's one of the questions biologists have had to deal with. And as they do that, they found that the DNA is constantly got all of these proteins around it. So this part here is the gene of interest. But to turn that gene on, every little shape that's in this figure is another protein that has to be bound to the upstream part of the DNA in order for that DNA to be translated or transcribed, excuse me, be transcribed into RNA. All right, so there's lots of proteins that bind and regulate this. And part of that regulation is that this end curves over, basically it bends, and this box here is this box here. And these regulatory proteins have to basically bind to it and to make that happen before you can get any RNA made. And this particular protein right here, whose name I don't really care, you know, is one of those proteins that got phosphorylated with synaptic transmission. So this is how you turn on the genes for strengthening synapses, when you had the calcium go up and turn on those protein kinases, and it went all the way back to the nucleus, and it can turn on a gene. All right, so we've got all these regulatory proteins. Well, some of the regulatory proteins like this come from synapses, and some of these regulatory species are other hormones as well. All right. But the real question of these, how do we get the right gene out of this knot when we need it, is the regulation of taking that chromosome and unwinding it when needed. And we call that epigenetics, the control of the genetic process. So it's not in the sequence of the DNA, which is you get half from your mom and half from your dad, but it's in the control of when that DNA gets unwound, and in order to make it work. And it's a set of chemical reactions that turns it on and off at specific times. And I don't know if you went to the Learn Genetics site and read the Lick Your Rats, but that's a fun one. Okay. But it turns out the DNA not only gets twisted, but it winds around other proteins in a slightly more ordered way, just like you'd wind your garden hose around one of those spools. Only it winds around multiple spools. Because it's so long, one spool isn't enough. And the proteins that form the spools are diagrammed here at the top, these balls with tails. So there's 8 of them, so they form, something that looks exactly like the spool for the garden hose. And the DNA, which is here, the light twisted thing, gets wound around the histones a couple of times. And then they go wind around another one, and then wind around a third or a fourth. And at the tips of these histone tails, you either get a methyl group or an acetate group biochemically. And which one is there determines whether that particular DNA wound around that particular histone gets unwound or not. So basically, the acetate says go, and the methyl says stop, stay wound. Okay? And so here in this picture at the bottom, you can see lots of acetates all over the histones, and you can see that the DNA is basically unwound from the very concentrated knot on the left. But if I methylate the DNA on the left, then this unwinding process won't happen.
Do you have wondered, do you have changed, I know there's chemicals inside with valence electrons. Do you have things that they establish connections? They change, like they change the molecules?
Well, there's enzymes that put on the acetate, and there's enzymes that take off the acetate, and there's enzymes that put on the methyl, and there's enzymes that take off the methyl.
Change actually connections and cause?
Well, not connections. The DNA strands, we want them to stay together. We don't want them to break. We have other enzymes that repair them if they break, but the idea of storing them so precisely is that they don't break, that we keep them in good shape and only unwind them when we need them. Okay.
So actually, acetylated or unacetylated, acetylated is a better condition because they are...
Well, acetylated means it's unwound and able to be transcribed. Okay. Methylated means stuck closed.
Oh, okay, so methylation is bad.
Well, it depends. Sometimes you don't want something turned on. Maybe you don't want a stress protein turned on if you can handle it, if it's down on the lower side of that U-shaped dose response curve. It really, everything in biology's got a plus side and a minus side. There's always a valence here. So we can't just say one is better. You have to be very careful here, all right? So guess what? All of our favorite proteins that go into making LTP can be regulated by these epigenetic controls on the DNA. And if you've got a lot of gene expression in a young kid, you get a lot of LTP, right? If you have no gene expression, then you might only have a transient potentiation and not something that lasts a long time. And basically, in this particular diagram, the unwinding isn't all that big. They're just showing a small section unwound. But it's unwound enough that this pentagram, this green pentagonal structure represents a protein that basically is turning on transcription. So it unwinds enough at the right place so that you can get something transcribed.
And what does transcribe mean?
That's where you're taking it from the DNA to the RNA. So you have to go from DNA to RNA, RNA to protein. Okay. So basic high school biology names you've forgotten, and they're easily mixed up, and I understand that. Okay. It's just that there's all of biology is all of this regulation. You think of remember the toys in your kid's bedroom when they were young enough to play but not put away? Everything went on the floor and it was chaos. That's called entropy. And that's what life would be if everything just went crazy. But in order to keep the inside of the cell orderly, there's all of these mechanisms that put the right proteins together at the right time and keep them stored where they should be until the right time. And so the whole cell is there to keep chaos from happening. And we're working against chaos at the molecular level here.
So we have to read these thick books about how things work out, right?
Or you can hope I talk faster. All right, so these epigenetic changes can vary by part of the brain and by time. So, this just says that, the different parts of the brain, the hippocampus, here's a long list of proteins that are regulated epigenetically, and here's a different list of proteins in the prefrontal cortex that are regulated epigenetically. And it doesn't matter that you know the names, but the hippocampal ones are all regulated within a day after whatever learning paradigm this particular investigator used, whereas the cortical ones kept being regulated for the next month. So the turning on and off of genes and when they are on and when they are off at these different levels can take a whole different kind of a time course. All right, so what does having epigenetics have to do with learning, memory, and trauma? Okay, all of my favorite proteins in synaptic plasticity are controlled epigenetically at synapses. So all of the good, synaptic plasticity is dependent on those. And you need the turning on of genes during a learning experiment. If you do it once, it makes it easier to do it again because of these long time spans. And if you turn on some of these genes during a traumatic experience, it makes those synapses extra strong and very hard to forget. Okay, not only do you have the association of lots of synapses working together, but you've got all this epigenetic stuff saying stay turned on. Crank this protein up and crank that protein down and just go full steam ahead, which can be very stressful. All right. And the other piece about this epigenetic regulation means that the phenotypes can be passed on to future generations. That's pretty special. But now, I'm done with the aside, and we're back to the story. All right, we're back to the story of dams and their pups. So in this experiment, the dams that licks her pups, as soon as her pups are born, they're switched with the pups from the mom who doesn't lick their pups. So if the pup licking behavior causing the change in the glucocorticoid receptors was followed the sequence of the DNA and the same kind of inheritance as blue eyes and blood type, then you would expect these pups to have a lot of glucocorticoid receptor, despite the fact that they had poor mothering from the non-liquor, anxious mom. And vice versa, you would expect these pups to have low high glucocorticoid receptor. Because it came from their mom, but that's not the case. It follows that the glucocorticoid receptor follows the behavior of the mom. It doesn't follow the genetics from the sequence of the DNA. And it has to do with the fact that the licking changes the level of methylation on the gene that controls the glucocorticoid receptor. So the level of your response to stress depends on how you were treated as a pup, if we're talking rats. And it also, as the glucocorticoid receptor in humans levels appears to be regulated by adverse childhood experiences and early life stress. There was a study done of brains from late teen, early 20s suicide victims who had had a history of adverse childhood experiences versus suicides without adverse childhood experiences. And the glucocorticoid receptor levels correlated with the adverse childhood experience. So, yes,
So what happens in the third generation?
it gets weaker. The experience has to be somehow or another reinforced from the second generation. But, I don't have a slide of this, consider a pregnant mom. Very early in pregnancy, that embryo is differentiating its cells into the different organs. And two of the organs that get formed very early on in embryogenesis are the gonads, the ones that are going to be reproductive organs of the male or female offspring. So the stem cells that are going to birth the third generation are basically being born during that first trimester, maybe even during the first 6 weeks. I'm not here, I don't know my weeks of gestation real well. Before the mom even knows she's really pregnant, or believes she's pregnant, or wants to admit she's pregnant. All right? And so basically, the levels of stress that a mom experiences during pregnancy can be passed on not only to her own first generation offspring, the F1 generation, but also to the grandchildren because of influences on those stem cells that later become the eggs or the sperm. So one pregnancy has the ability to affect 2 additional generations. So it's pretty, I hate to say this to young ladies, but when you're pregnant, go to the South Seas. I mean, stay away from any possible stresses you can. Yeah, I know. I mean, because you don't know. We stress ourselves all the time. We have excess liquor consumption. We smoke. We have all kinds of behaviors, and it's not one behavior. It's probably cumulative multiple behaviors. And my story gets even worse. All right. Ready for the next chapter? You got more questions here. All right. So here's just some examples of changes in early life stress in a bunch of species where they're looking at maternal separation. This is actually humans here, and this is EEG data projected onto a circular idea of the brain from Romanian orphans. So this was a generation ago, basically. If the child got into foster care before they were 24 months old, then there was plenty of EEG activity throughout the brain. But if they did not, if they were institutionalized in those horrendous conditions, then there was very little EEG activity. So that says something about circuit formation in the brain of children who were neglected. This is a monkey study where the monkey has been deprived of its mom for either a week or a month, and here's a control animal. And they're looking for expression of this particular enzyme, which is a part of energy metabolism. And it just shows you that for the first week of after the mom was taken away, the stress is so acute that there's less energy generated, and then there's a rebound a month later, there's more of this energy. So energy enzyme that's expressed. So there's an oscillation here. The body is trying to respond, and it's very hard to predict. It's not just a simple on or off, but it's an oscillation that's going on. If you look in, again, I think this is a rodent, but I honestly don't know what the species is here in D. If there's this maternal separation for a week in infancy, you can see underneath there we go. You can see here that there's fewer dendrites, fewer branches to the dendrites, so less complex neurons, not getting as much input. But if that happened in childhood as opposed to infancy, now you've got more branches on the dendrites. Again, a paradoxical change we can't totally explain, but the body is trying to compensate, and it is doing something different. All right, we know it's not normal. And it will change because we don't even know what neurons these are. We don't know if they're excitatory or inhibitory. So if they're inhibitory neurons with more dendrites, that could be less activity. We don't have good, every technique doesn't tell us all the answers. But there is a decrease in inhibitory neurons in the development, and they develop very shortly after birth. So this kind of maternal separation early in life can actually close what we call a critical period. And that's what's diagrammed here, is that the normal critical period for learning language or learning phonemes or whatever is shifted earlier in life. It closes sooner. So if there's plasticity, it's still there, but it doesn't extend to the ages that we might want it to extend to. It might be over before the kid gets to kindergarten. Okay, and it depends on the critical period for what. But this change in the balance of neurons really has a strong effect on development and growth of the offspring. All right. So the glucocorticoid receptor's got epigenetic controls. What about this feedback protein, FKBP5? All right, turns out FKBP5 has two different forms that show up in the human population. One form predisposes you to resilience. The other form predisposes you to high risk for stress-related disorders.
And what determines which one you get?
Whether there's an A and a T at this particular site.
I want to know which one I got.
A G or a C. And don't ask me what the frequency is. I don't know what the frequency is, and I don't know what, I can't tell you it's 50-50 or 25-75. I don't know that.
So what determines which one you get?
It's just you inherit it. This form is inherited. This is in the sequence itself. It's not in the epigenome. It's in the sequence itself. But the FKBP5 protein is going to have epigenetic regulation as well. So not only will it have a difference in sequence for some people, but it will have epigenetic regulation that depends on experience. Mm-hmm. So if you have the high-risk genotype and you are in harm's way, there is great likelihood that with accumulation of life stress, over time, you will increase your stress response, and you can increase your FKBP5 and turn off some of these feedback loops that regulate the stress response, which keeps the stress response up longer, and you can then become subject to long-term stress-related problems, which we would call psychiatric illness or aberrant behaviors or some of these other kinds of pathologies that accrue to people who have been in trauma. Whereas other people who have the resilient genotype exposed to the same trauma might be able to understand it, manage it, and move on in life. And it will be both a function of the flavor of the FKBP5 protein they were born with, and the experiences that methylated or didn't methylate their DNA.
Are there like experiments that people have been able to see whether people who have developed like PTSD tend to have more of the high-risk genotype? Have they been able to
That's a question for Brian. I don't
correlate
know the answer. That's you
now they
really
know one thing to ask them.
you really are now at the edge of my knowledge, all right? Because I had to read this in order to give you this connection. But this is basically where I wanted to be. Sorry it took me
That
so
might be
long.
at the end of his
Interesting.
talk.
He was talking about a
He was
different
talking.
system. He was talking about a different system. He was talking about interleukins, which are not cortisol, but they're a long-term. The level of interleukins says long-term how much stress you've had cumulatively. So it's a different molecule that's measuring accumulation as opposed to dynamic change. But in order to understand that cumulative, you kind of needed to know this other. And that's where I felt like you weren't able to appreciate the complexity, because he's talking about molecules that not only link nervous systems and learning and memory, but link immune systems and whole other body systems. So he's taken these and he's gotten us to the whole body piece of this, as opposed to just the brain part of this. And I know some of the brain part of it. So basically, we're back to my summary slide at the beginning. I'm really done. It's not nature or nurture, it's nature and nurture. Your experiences are really, 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 really important. But we can't all protect our kids forever and ever. Everybody goes out into the world, so we have to be able to manage these stresses in whatever forms they accumulate in our lives, and realize that they need managing. And it's not a done deal. We don't know what stress is coming to any of our lives tomorrow. But we have to be able to think about the complexity of this. And when it comes to treatment, what would the treatment be different if you knew what the FKBP5 allele was that you had? I don't know. I don't think we have treatment that's that refined at this point. But I honestly don't know. I'm not a clinician. I'm not fuddy-duddy.
So it means you have an offspring and you can manipulate, try to manipulate and say, avoid the stress now, or now you need the stress to improve, right? You don't know.
You don't, yeah, you always want enough stress that the child is motivated. All right, you can't sit
Too
around and be a
much,
couch potato the whole time. And you can't let them run wild like a banshee. So you really have to, we have to think hard about these issues. But would we change our parenting? I don't know. I mean, good parenting is still good parenting. What's the equivalent to licking your pups? Touching and embracing and interacting with offspring. And those are really young reactions. You lick the pups before they're weaned. And so that's, you lick the pups before their eyes are open. A lot of rodent gestation happens after birth. But, in human, we carry much longer, and so a lot more of that development happens in utero. But we still need that emotional interaction, certainly. Maternal absence is huge, and that's a huge stressor. But, other adverse childhood events, poverty, immigration, a parent who's in prison. I mean, there's lots of other ways that we can stress kids.
So it's not a chronological thing, you could say at this time this happened. You don't know, because that may be different by different...
And it's very hard in human populations to say, this was the event that caused schizophrenia 20 years later. But you can add up adverse childhood experiences, and 4 or more will predict poorer outcomes. And that's irrespective of the FKBP5 allele, whether you're resilient or not resilient.
And if you could measure it, and you have a system that you measure, okay, now is the time to put the stress, now it's not.
No.
You cannot do that.
But would we want to? Would we want every kid raised identically according to some timetable? I mean, that sounds pretty Orwellian to me.
Would this difference in like the FKBP5, would this be something that could explain why maybe like siblings who go through the same experience, one of them is much more traumatized than the other?
Absolutely could. It could also mean that the parent responded differently to the boy than the girl. It's,
Okay,
it's,
there's so many.
there's a lot. Because the one thing I didn't show you, guess what else interferes with all of this? Sex hormones, estrogen, testosterone, they bind the same places that glucocorticoids bind. So you're going to get a different response based on sex in a lot of this. Cortisol levels vary between men and women differently. So, there's all of those different colored blobs are different proteins doing different things, and some of them are estrogen and testosterone or androgens. I mean, so it's much more complex. But everything you can think of having an effect will. Sorry. So, it's hopeful. I mean, we like to think of synaptic plasticity as hopeful, that there is this ability to recover. And if we just encourage it enough, can we overcome some of these stress-related issues? But it does help to know the physiology, yeah.
Most of these things are produced by your body, but are there cortisol and things like that be produced by pharmaceuticals that are given to people to help change those stress levels?
There's synthetic ones. Dexamethasone is one that's used in some conditions for some hormone related abnormalities, because you have a problem with your adrenals or your pituitary, and those levels can get off, sure. You have tumors that secrete hormones on their own. Yeah, there's lots. I mean, that's what you go to an endocrinologist for. I can't diagnose, I can't tell you what those are. You're really edging my knowledge.
I'm not saying that.
No. But I just, I can show diagrams. I can't do more than that. I talk way too long.
No.
Perfect.
So stand up and stretch. I gave you a
What will
lot.
that do?
Well, to wake you up. I gave you a lot to think about.